Sealed cave awakens. Movile Cave was discovered in Romania after workers opened a hidden shaft during construction surveys.
Sealed cave breathes. Behind of entrance lay a sealed underground world, cut off from sunlight for millions of years. Sealed cave survives. Cave air contains low oxygen and unusually high carbon dioxide, methane, ammonia, and hydrogen sulfide. Sealed cave feeds. Those gases would be dangerous for people, and yet they help power an entire isolated ecosystem. Sealed cave evolves. Unlike forests or reefs, this food web begins without sunlight, leaves, algae, or photosynthesis today. Sealed cave waits. Microbes oxidize chemical fuels from groundwater, turning toxic compounds into biological energy and biomass today. Sealed cave thrives. This process is called chemosynthesis, where life uses chemical reactions instead of sunlight for energy. Sealed cave hides. Floating microbial mat forms the base of the food chain in the cave's sulfidic water. Sealed cave forms. Small invertebrates graze on bacterial films while predators hunt them through permanent darkness underground today. Sealed cave deepens. Movile Cave turns poison gas into hidden ecosystem, making it one of Earth's strangest habitats. Sealed cave persists. Cave remained naturally sealed until its accidental discovery, limiting outside airlight and organic matter. Sealed cave glows. Isolation allowed unique species to evolve in a stable but hostile chemical environment underground today. Sealed world awakens. Researchers enter carefully because a human breath can change the cave's fragile atmosphere and humidity. Sealed world breathes. Access is restricted to protect both scientists and organisms adapted to extremely specific conditions today.
Sealed world survives. The entrance shaft descends into limestone passages where normal daylight biology disappears almost completely today.
Sealed world feeds. Every surface seems ordinary at first, but chemical gradients shaped the hidden living world today. Sealed world evolves. The water smells sulfurous because hydrogen sulfide rises from deep thermomineral groundwater below underground today. Sealed world waits. Methane and ammonium also feed microbial metabolism, adding more chemical pathways to the ecosystem today. Sealed world thrives. Oxygen is scarce, yet thin oxygenated boundaries allow sulfur oxidizers to build biomass underground today. Sealed world hides. That boundary between oxygen and sulfide becomes the engine room of the cave ecosystem today.
Sealed world forms. Bacterial mats spread across the water surface like pale living blankets in total darkness today.
Sealed world deepens. Their cells harvest energy by stripping electrons from reduced chemicals dissolved in groundwater underground today.
Sealed world persists. The microbes convert carbon dioxide into organic matter, creating food without any plant leaves today. Sealed world glows. This makes the cave resemble deep-sea hydrothermal systems more than ordinary limestone caves underground today.
Sealed water awakens. In both places, chemistry replaces sunlight as the foundation of a hidden food web today. Sealed water breathes. The mat supports springtails, isopods, snails, leeches, spiders, pseudoscorpions, and other cave animals underground today. Sealed water survives. Many species are endemic, meaning they are known only from this single cave system today. Sealed water feeds. Several animals lost pigment because color provides little advantage in permanent darkness there underground today. Sealed water evolves. Some reduce their eyes, relying more on touch and vibration, smell, and chemical cues underground today. Sealed water waits. Evolution reshaped ordinary arthropod body plans for an environment without day, night, or seasons today. Sealed water thrives. A blind water scorpion lives in the cave waters hunting without ordinary visual guidance today. Sealed water hides. Predatory cave spiders and pseudoscorpions patrol dry passages searching for prey in chemical darkness underground today. Sealed water forms. Leeches and small crustaceans move through microbial films where poison gases feed their food source. Sealed water deepens. The animals do not eat gas directly, and they depend on microbes converting chemicals first today. Sealed water persists. Food relationships form a ladder, from gas-fed bacteria to grazers and then predators underground today. Sealed water glows. Stable isotope studies revealed that cave animals carry chemical signatures from chemoautotrophic microbial production today. Sealed air awakens. That evidence showed the ecosystem's foundation is inside the cave, not washed from outside today. Sealed air breathes. Small amounts of outside material may enter, but they do not explain main food web. Sealed air survives. The cave became famous because it proved complex land ecosystems can persist without sunlight today. Sealed air feeds. It also challenged assumptions about where life can survive on Earth and beyond underground today. Sealed air evolves. The atmosphere can contain several percent carbon dioxide far above ordinary outdoor air underground today. Sealed air weights. Low oxygen makes the cave hazardous, requiring careful planning and controlled scientific access underground today. Sealed air thrives. Hydrogen sulfide can disrupt breathing chemistry, and so the cave is not a tourist attraction today. Sealed air hides. Researchers use protective procedures to avoid poisoning, contamination, and disturbance of fragile habitats underground today.
Sealed air forms. The cave's danger is chemical and not supernatural, and it is not filled with monsters today.
Sealed air deepens. Its alien feeling comes from biology adapting to real geochemical energy sources there underground today. Sealed air persists. That distinction makes Movile Cave more interesting than a fantasy horror setting there underground today. Sealed air glows. Every organism survives inside limits shaped by temperature, humidity, oxygen, sulfide, and food underground today. Sealed ecosystem awakens. A small change in ventilation could shift the chemistry that supports the ecosystem underground today. Sealed ecosystem breathes. Protecting the cave means protecting chemical balance, and not just protecting rare animals there underground today. Sealed ecosystem survives. Limestone formed the cave through groundwater dissolving carbonate rock over long geological timescales underground today. Sealed ecosystem feeds. The surrounding region sits near the Black Sea coast, but the cave's water is isolated. Sealed ecosystem evolves. Deep aquifers bring warm, mineral-rich water upward through fractures beneath the landscape there underground today. Sealed ecosystem waits. Those fluids deliver sulfide methane and ammonium that microbes can use as fuels underground today. Sealed ecosystem thrives. Where groundwater meets limited oxygen, microbial chemistry builds the cave's living foundation there underground today. Sealed ecosystem hides. The same chemical interface creates milky biofilms and floating mats, visible under researchers' lamps today. Sealed ecosystem forms. Without that interface, animals would lose the food source sustaining their community there underground today. Sealed ecosystem deepens. The cave demonstrates how geology, water, and microbes can cooperate to build ecosystems underground today. Sealed ecosystem persists. Its sealed state preserves an unusual natural laboratory for studying evolution under isolation underground today. Sealed ecosystem glows. That laboratory helps scientists ask how life adapts when sunlight disappears entirely there underground today. Sealed passage awakens. Genetic studies reveal lineages diverging from surface relatives after long underground isolation there underground today. Sealed passage breathes. Some cave species arrived at different times, so the ecosystem was assembled in stages today. Sealed passage survives. Adaptation did not happen instantly, and populations changed gradually. Sealed passage feeds. Blindness, pale bodies, long appendages, and slow metabolisms reflect different survival pressures there underground today. Sealed passage evolves. Predators still exist, proving darkness does not eliminate ecological conflict underground below there underground today. Sealed passage waits. Competition also occurs among microbes as different species exploit sulfide methane or ammonia underground today. Sealed passage thrives. Microbial communities recycle sulfur, carbon, nitrogen, and other elements through connected chemical loops underground today. Sealed passage hides. Those loops keep nutrients moving even when surface leaves and sunlight never arrive underground today. Sealed passage forms. The cave becomes a miniature planet where chemistry writes the rules for life underground today. Sealed passage deepens. Scientists study it to understand early Earth environments where sunlight was not always central today. Sealed passage persists. Movile Cave also inspires comparisons with icy moons that may hide oceans below crusts today. Sealed passage glows. Europa and Enceladus are not the same, but chemical ecosystems make the analogy useful today. Sealed darkness awakens. If life can use rock-derived energy here, similar principles might matter elsewhere there underground today. Sealed darkness breathes. That does not prove alien life exists, but it expands scientific imagination responsibly underground today. Sealed darkness survives. The cave teaches that sunlight is powerful, yet not the only possible biological engine today. Sealed darkness feeds, deep-sea vents, sulfidic caves, and subsurface aquifers all reveal alternative energy pathways there underground today. Sealed darkness evolves. Movile Cave stands out because its ecosystem is accessible enough to study on land today. Sealed darkness thrives. Researchers must balance curiosity with conservation every time they open the entrance there underground today. Sealed darkness hides. Sampling can remove organisms, change humidity, introduce microbes, or disturb delicate bacterial mats underground today.
Sealed darkness forms. Modern studies use careful sequencing microscopy chemistry and stable isotopes to reduce uncertainty underground today.
Sealed darkness deepens. No single method explains everything, so evidence must connect molecules and species and environment underground today. Sealed darkness persists. Cave maps reveal dry chambers, flooded passages, air pockets, and water-filled tunnels there underground today. Sealed darkness glows. Some corridors are cramped, humid, and slippery, making exploration physically difficult and risky underground today. Sealed microbes awakens. Lighting must remain controlled because these organisms evolved without natural light exposure there underground today. Sealed microbes breathes. Even a researcher stepping through mud can disturb microbial films and tiny animals underground today. Sealed microbes survives. The cave is therefore both a discovery site and a protected biological experiment underground today. Sealed microbes feeds. Its story begins with accident, but its scientific value depends on restraint there underground today. And sealed microbes evolves. Clickbait headlines call the inhabitants alien, but they are Earth organisms with extreme adaptations today. Sealed microbes waits. They look strange because evolution solves problems differently when sunlight vanishes underground there, underground today. Sealed microbes thrives. The real monster is chemistry, invisible gases, low oxygen, and suffocating air there underground today. Sealed microbes hides. Still, those same dangerous chemicals support the microbes that feed the cave community underground today.
Sealed microbes forms. Poison and food become two sides of one biogeochemical system inside Movile Cave underground today.
Sealed microbes deepens. That paradox makes the cave scientifically powerful and visually unforgettable for storytelling there underground today. Sealed microbes persists. A sealed cave with no sunlight should seem lifeless by ordinary intuition there underground today. Sealed microbes glows. Instead, it reveals a crowded ecosystem built from sulfur, methane, and microbial chemistry underground today. Sealed chamber awakens. The animals are not fossils, and they are active participants in a living underground web today. Sealed chamber breathes. Every grazer, predator, and bacterium depends on invisible chemical gradients in the darkness underground today. Sealed chamber survives. The cave also reminds viewers that Earth's hidden biosphere is larger than surface life suggests.
Sealed chamber feeds. Underground habitats may hold many species still unknown to science today below there underground today.
Sealed chamber evolves. Some discoveries are tiny, pale, eyeless creatures rather than dramatic ruins or treasure underground today. Sealed chamber waits. Their importance comes from showing how life persists under constraints that seem impossible underground today. Sealed chamber thrives. Scientists continue cataloging species genomes and metabolic pathways inside the cave below there underground today. Sealed chamber hides. Each new finding refines the picture of how isolated ecosystems organize themselves there underground today. Sealed chamber forms. The cave's chemistry can shift slowly, so long-term monitoring matters for understanding stability underground today. Sealed chamber deepens. Climate, groundwater movement, and human activity could influence conditions even in sealed systems underground today. Sealed chamber persists. That makes conservation urgent, because rare ecosystems can be damaged before they are understood today.
Sealed chamber glows. Movile Cave is not a place for adventure tourism or careless exploration there underground today.
Sealed laboratory awakens. It is a biological vault protecting evolutionary experiments conducted by time rock and chemistry today. Sealed laboratory breathes. The darkest chambers show that life does not need sunlight to be complex underground today. Sealed laboratory survives, and they show that energy, water, chemistry, and time can build unexpected worlds there underground today. Sealed laboratory feeds. A poisonous cave becomes a lesson about resilience, adaptation, and planetary possibility there underground today. Sealed laboratory evolves. That lesson changes how we imagine life's limits on Earth and beyond there underground today. Sealed laboratory waits. When the shaft closes, the cave returns to darkness, humidity, and chemical silence underground today. Sealed laboratory thrives, but inside that silence, microbes keep turning poison gases into the foundation of life today. Sealed laboratory hides. Movile Cave reveals a hidden world where the sun never rises, yet ecosystems still breathe. Sealed laboratory forms. Its strangest truth is simple. Earth's alien worlds can exist right beneath our feet today. Sealed laboratory deepens. Story ends underground, where poisonous gases become the first step in a living food chain.
Sealed laboratory persists. Sunlight never enters, but chemical energy keeps moving through microbes, grazers, and predators underground today. Sealed laboratory glows. That is why Movile Cave feels impossible while remaining completely grounded in science underground today.
This Antarctic fish reaches adulthood without red blood cells, leaving its circulating blood pale and nearly transparent. Its entire evolutionary story unfolded within the isolated, freezing waters surrounding Antarctica. The Antarctic circumpolar current helped isolate southern fishes from warmer oceans for millions of years, supporting southern ocean separation. These seas remain cold enough to freeze the body fluids of most unprotected fishes, central to sub-zero seawater exposure. Antifreeze glycoproteins prevent dangerous ice crystals from growing inside many Antarctic nodovoid fishes, central to antifreeze crystal control. These proteins attach to ice and interrupt the repeating structure needed for further growth, supporting ice-binding molecular defense. Cold water slows biochemical reactions, movement, digestion, and nearly every other process inside the animal, supporting cold-slowed biochemistry. A comparatively low metabolic demand reduces the amount of oxygen its tissues must receive each minute, supporting reduced metabolic demand. Nearly every other vertebrate transports oxygen using red cells packed with hemoglobin, while preserving standard vertebrate oxygen transport. Hemoglobin multiplies blood's oxygen capacity by reversibly binding gas molecules inside circulating cells, which affects hemoglobin oxygen binding. Icefish blood lacks both the red cells and their dominant respiratory pigment, sustaining translucent plasma circulation throughout Antarctic activity. Under a microscope, its circulation appears strikingly different from ordinary vertebrate blood, which affects erythrocyte-free blood samples. Oxygen must travel physically dissolved within plasma rather than attached to hemoglobin, sustaining plasma-dissolved oxygen throughout Antarctic activity. That method carries only a small fraction of the oxygen transported by red-blooded relatives, supporting weak oxygen delivery. Losing hemoglobin reduced blood oxygen capacity by roughly an order of magnitude, with consequences for collapsed carrying capacity. Yet the fish survives because its entire circulatory system compensates for that enormous deficit, supporting whole system compensation. No single adaptation explains life without red blood. Several mechanisms work together continuously, which cold seawater naturally holds more dissolved oxygen than warm seawater, compensated by cold water oxygen abundance inside Antarctic tissues. Abundant environmental oxygen improves diffusion across the gills, despite weak transport inside the blood, which affects gill diffusion efficiency. The Antarctic environment made a normally catastrophic blood defect temporarily survivable. Adult crocodile ice fishes are the only known vertebrates completely lacking circulating red blood cells, central to adult erythrocyte absence. Their developmental program no longer produces the mature oxygen-carrying cells typical of vertebrates, supporting arrested blood cell development. Genomic evidence records the breakdown of genes required for functional hemoglobin, compensated by globin gene degeneration inside Antarctic tissues. Key globin genes were deleted or reduced to non-functional genomic remnants, compensated by non-functional genomic remnants inside Antarctic tissues. Globin loss occurred after the icefish lineage separated from hemoglobin-producing notothenoids, while preserving post-divergence hemoglobin loss. Modern white-blooded icefishes inherited the condition from a common ancestor, compensated by shared ancestral deletion inside Antarctic tissues. The family later diversified while retaining its unusual blood physiology, compensated by icefish family diversification inside Antarctic tissues. Myoglobin followed a different evolutionary path and disappeared independently in several icefish species, which affects independent myoglobin loss. Where present, myoglobin helps buffer oxygen inside constantly active heart cells, revealing cardiac oxygen buffering under stable cold conditions. Some icefish hearts function without either hemoglobin or cardiac myoglobin, sustaining myoglobin-free heart function throughout Antarctic activity. Independent losses demonstrate that similar physiology evolved repeatedly within the family, compensated by repeated evolutionary loss inside Antarctic tissues.
Scientists still debate whether hemoglobin loss was advantageous or merely survivable under extreme cold.
Removing red cells made blood easier to pump while severely weakening oxygen transport, which affects viscosity-capacity trade-offs. Lower viscosity reduces resistance within the circulation, partly offset through low-resistance plasma flow across cold tissues. Greater cardiac output partly offset the small amount of oxygen carried per unit blood, which affects elevated cardiac output. Wide vessels allow high-volume flow without excessive pressure, partly offset through wide vessel circulation across cold tissues. Dense capillary networks shorten the distance between plasma oxygen and tissue cells, tied closely to dense capillary architecture. Mitochondrial abundance increases the cellular surface available for oxygen use, partly offset through mitochondrial oxygen use across cold tissues. Icefish mitochondria also display unusual density, size, and internal organization, partly offset through unusual mitochondrial morphology across cold tissues.
Hemoglobin loss reshaped anatomy from genes to vessels, heart, and cellular energy machinery, especially through genome-to-heart compensation.
Every delivered oxygen molecule begins as gas dissolved within the surrounding seawater, while preserving gill plasma oxygen transfer.
Lower temperature increases the amount of oxygen water can hold, compensated by temperature-dependent oxygen solubility inside Antarctic tissues. Polar conditions therefore maximize the oxygen available for passive uptake, compensated by polar passive uptake inside Antarctic tissues. Thin exchange surfaces reduce the distance oxygen must diffuse, partly offset through thin membrane diffusion across cold tissues.
No cellular carrier captures the gas after it enters circulation, revealing carrier-free circulation under stable cold conditions.
Carrying more plasma increases the total dissolved oxygen moving through the animal, sustaining expanded blood volume throughout Antarctic activity. Icefishes maintain substantially greater blood volume than many related fishes, partly offset through high plasma volume across cold tissues.
Cell-free blood flows with comparatively low resistance, partly offset through low cellular crowding across cold tissues.
High vascular density increases the area available for oxygen delivery, compensated by vascular oxygen delivery area inside Antarctic tissues. Short diffusion distances compensate partly for low oxygen concentration, partly offset through short tissue diffusion across cold tissues. Delivery succeeds through broad distribution rather than concentrated oxygen cargo, partly offset through distributed capillary delivery across cold tissues. Behavior also keeps oxygen demand within the circulation's restricted capacity, partly offset through behavioral oxygen conservation across cold tissues. Intense activity can challenge a system with little oxygen reserve, partly offset through burst swimming limitation across cold tissues. Icefish generally possess limited scope for prolonged high-performance swimming, partly offset through low aerobic scope across cold tissues. Warmer conditions raise metabolic demand while lowering water's oxygen content, compensated by warming-driven oxygen mismatch inside Antarctic tissues. Temperature therefore compresses the animal's physiological safety margin from both directions, compensated by shrinking physiological margin inside Antarctic tissues. Its unusual blood works best within the environment where it evolved, compensated by stable sub-zero performance inside Antarctic tissues.
And local circulation can provide relatively stable thermal conditions, compensated by deep water thermal refuge inside Antarctic tissues.
Shallow populations may encounter faster environmental change through shallow water exposure, limiting stress while sustaining water exposure.
Environmental oxygen and cardiovascular compensation remain inseparable parts of the same survival system, which affects environment circulation coupling. Icefish hearts are enlarged relative to those of similarly sized red-blooded relatives, which affects enlarged ventricular anatomy. Each beat moves an unusually large volume through the circulation, supporting high stroke volume throughout Antarctic cardiac activity. High stroke volume raises cardiac output without requiring extremely rapid heart rates, with consequences for cardiac output compensation. Old cardiac muscle contracts slowly and depends on specialized cellular machinery, compensated by cold cardiac machinery inside Antarctic tissues. Mitochondrial density supports aerobic energy production inside the oversized heart, partly offset through heart mitochondrial density across cold tissues. Dense vessels keep oxygen pathways short within cardiac tissue, partly offset through short cardiac diffusion across cold tissues. Some species add myoglobin storage within the heart, partly offset through retained cardiac myoglobin across cold tissues. Other species survive after losing cardiac myoglobin independently, partly offset through independent myoglobin absence across cold tissues. Different lineages reach similar physiological outcomes through distinct genetic changes, partly offset through convergent cardiovascular outcomes across cold tissues.
Elevated nitric oxide signaling may help promote wide vessels and vascular growth, tied closely to nitric oxide signaling.
Nitric oxide regulates both blood flow and formation of new vessels, compensated by angiogenic vessel growth inside Antarctic tissues.
Long-term signaling can reshape anatomy throughout growth. Through developmental compensation,
Vascular differences emerge as the animal develops, through heart growth trajectories, limiting stress while sustaining heart growth trajectories in Antarctica.
static signals may transform a blood defect into a stable body plan, sustaining homeostatic circulatory remodeling throughout Antarctic activity. Low viscosity makes large volume pumping mechanically easier, partly offset through viscosity-enabled high flow across cold tissues. Tips. Cardiovascular compensation still requires substantial energy. Through energetic compensation costs, limiting fish pays for weak blood with a larger circulatory apparatus, revealing large circulatory investment under stable cold conditions. Warmer water can push that enlarged system toward its performance ceiling, sustaining thermal cardiac limits throughout Antarctic activity. Cardiac capacity may limit acute warming tolerance in some species, partly offset through cardiac performance ceiling across cold tissues. Every compensation traces back to an ancient loss recorded in the genome, with consequences for integrated polar physiology. Their closest red-blooded relatives still retain that ancestral system, partly offset through retained relative physiology across cold tissues. A major deletion destroyed essential hemoglobin coating material, partly offset through beta-globin deletion across cold tissues. Additional remnants preserve a molecular fossil of the lost pathway, compensated by alpha-globin genomic fossil inside Antarctic tissues. Once the protein was no longer required, disabling mutations accumulated more freely, central to post-loss mutation accumulation. Blood physiology changed far beyond the globin genes themselves, partly offset through reduced iron physiology across cold tissues. White blood contains dramatically less iron bound in respiratory pigment, compensated by iron-poor white blood inside Antarctic tissues. The absence of red cells lowers viscosity substantially, partly offset through erythrocyte-free viscosity across cold tissues. Lower viscosity may have softened the damage without making hemoglobin loss beneficial, sustaining evolutionary trade-off interpretation throughout Antarctic activity. Environmental conditions created a rare window in which the mutation could persist, sustaining cold ancestral tolerance throughout Antarctic activity. Similar gene loss would be far less tolerable in warmer, oxygen-poor seas, which affects warm water incompatibility. Red-blooded relatives often retain greater capacity for oxygen transport during thermal stress, which affects relative thermal capacity. Extreme specialization can increase success in stable environments while reducing resilience to rapid change. Supporting specialization, genome studies reveal additional adaptations beyond blood and circulation, partly offset through antioxidant gene expansion across cold tissues. Oxygen-rich water and abundant mitochondria can increase oxidative stress, partly offset through mitochondrial oxidative pressure across cold tissues. Strong antioxidant and repair systems limit damage from reactive molecules, partly offset through macromolecular repair defense across cold tissues. Icefish survival depends on a broader Antarctic molecular toolkit, partly offset through broader cold adaptation across cold tissues. Traits optimized for constant cold may become liabilities during warming, partly offset through warming-created mismatch across cold tissues. Climate change modifies several ecological pressures simultaneously through multi-factor climate pressure. Risk will vary among species, locations, depths, and life stages, partly offset through species-specific exposure across cold tissues. Warm water holds less dissolved oxygen while animal metabolism accelerates, partly offset through warming oxygen decline across cold tissues. Icefish face simultaneous reductions in supply and increases in demand, compensated by aerobic supply-demand compression inside Antarctic tissues. Limited oxygen carrying capacity may bring some icefishes to their ceiling sooner, tied closely to earlier performance ceilings. Thermal vulnerability is not identical across the family, partly offset through variable family vulnerability across cold tissues. Behavioral movement may reduce exposure to warming surface layers, partly offset through behavioral thermal refuge across cold tissues. Mutagens. Suitable cold refuges may not exist everywhere. Through geographic restraints, limiting stress, while early life stages may encounter different thermal risks than adults, compensated by larval thermal exposure inside Antarctic tissues. Warming can reshape development before circulatory compensation is fully established, balanced through developmental oxygen stress during polar life. Changing prey fields can indirectly affect icefish energy budgets, partly offset through changing prey networks across cold tissues. Reduced food could make costly cardiovascular compensation harder to maintain, partly offset through cardiovascular energy deficit across cold tissues. Field monitoring is essential for separating short experiments from population trends, with consequences for long-term population monitoring. Research collections reveal anatomy, genetics, diet, and physiological condition, partly offset through research specimen collection across cold tissues. Modern tools connect whole animal performance with molecular mechanisms, partly offset through integrated molecular analysis across cold tissues. Comparative studies identify which traits followed hemoglobin loss and which evolved earlier, with consequences for comparative evolutionary mapping. Variation within the family turns icefishes into a natural evolutionary experiment, with consequences for within-family physiological variation.
Their biology offers insights into oxygen transport without providing a direct model for human treatment, supporting biomedical research boundaries.
Understanding compensation may illuminate general principles of cardiovascular plasticity, partly offset.
Those adaptations remain inseparable from the cold, oxygen-rich sea that made them possible, helping explain Southern Ocean dependence.
Its future depends on whether physiology can keep pace with changing Antarctic water, tied closely to future warming pressure.
Antarctic icefish survive without red blood by rebuilding nearly every other part of oxygen delivery, supporting integrated oxygen delivery. Our silent depths. Mariana Trench marks the deepest known part of Earth's ocean, stretching through western Pacific near Guam. Our silent waters. Its lowest area, Challenger Deep, drops farther below sea level than Everest rises above it. Our silent pressure. This trench forms where Pacific Plate dives beneath smaller Mariana Plate at a subduction zone. Our silent darkness. As one plate sinks and seafloor bends downward, creating a long curved scar across the ocean. Our silent trenches. From space, the trench looks invisible, but below waves it slices dramatically into Earth's crust. Our silent currents. Most of Trench lies east of the Mariana Islands, an arc built by tectonic collisions. Our silent seafloor. Scientists measure the deepest points carefully because pressure currents and terrain complicate every estimate underwater. Our silent abyss. Even today, exact depth values vary slightly depending on sonar methods, instruments, and calibration standards. Our silent silence. At the bottom, pressure exceeds 1,000 times atmospheric pressure felt at Earth's surface normally. Our silent worlds. That means every square inch experiences crushing force strong enough to destroy ordinary unprotected machines. Our silent shadows. Sunlight disappears long before the trench floor, leaving a black world without natural daytime illumination. Our silent frontiers. Temperatures stay cold, usually just above freezing, although geothermal activity can locally alter conditions somewhat. Our Hadal depths. Food is scarce this deep, so life survives on marine snow carcasses and microbial chemistry. Our Hadal waters. Marine snow includes dead plankton, fecal pellets, mucus, and drifting fragments sinking from brighter waters. Our Hadal pressure. Occasionally, a whale fall delivers a huge food bonanza to deep scavengers and bacteria below. Our Hadal darkness. The trench is not empty. It hosts amphipods, microbes, sea cucumbers, and Hadal snailfish today.
Our Hadal trenches. Hadal zones describe ocean depths deeper than 6,000 meters, including trenches and deepest troughs.
Our Hadal seafloor. Life here adapts through flexible proteins and specialized membranes and chemistry that resists extreme pressure damage. Our Hadal abyss. Some deep animals avoid gas-filled spaces entirely because compressible cavities fail under enormous surrounding pressure. Far Hadal worlds. Researchers found snailfish near the trench bottom, proving vertebrates can live astonishingly deep underwater today. Our Hadal shadows. These deep-sea snailfish appear delicate, yet their bodies contain molecules helping proteins keep functioning under pressure.
Far Hadal frontiers. Large amphipods roam the Hadal floor, scavenging whatever organic matter reaches the abyssal sediments below. Far shadowed depths. Xenophever's giant single-celled organisms can carpet muddy slopes and provide habitat for smaller animals today. Far shadowed waters. Microbes also thrive there, metabolizing chemicals and recycling nutrients within sediments hidden beneath darkness today. Far shadowed pressure. Some bacteria consume organic matter, and others exploit compounds released by rocks' fluids or decomposition below. Far shadowed trenches. Each layer of mud can record tiny fossils, ash, or contaminants delivered across decades today. Far shadowed currents. Scientists sample these layers like pages inside a buried archive of ocean and planetary history. Far-shadowed seafloor. Trench also matters for earthquakes, because subduction zones store stress and sometimes release devastating ruptures. Far-shadowed abyss. Nearby tectonic activity can trigger quakes and tsunamis, though not every trench segment behaves identically. Far-shadowed silence. Volcanoes west of the trench arise when water from the descending plate helps generate magma. Far-shadowed worlds. So the trench connects directly to the fiery island arc above, despite its frozen darkness. Far-shadowed shadows. This pairing shows how Earth's deepest ocean and volcanically active islands share one tectonic engine. Far-shadowed frontiers. Humans first reached Challenger Deep in 1960, aboard the bathyscaphe Trieste, a remarkable descent. Far abyssal waters. They reported a flat, silty floor, surprising many who imagined jagged cliffs and constant chaos.
Far abyssal pressure. Decades later, robotic vehicles returned with cameras, sensors, and samplers more capable than Trieste today.
Far abyssal currents. Their missions revealed ridges, sediment plains, escarpments, and unexpected debris left by human civilization below. Far abyssal seafloor, the deepest ocean, contains pollution, including plastics and chemicals, transported from distant surface activity. Far abyssal abyss. Scientists discovered plastic fibers and persistent toxins inside trench animals, showing contamination reaches astonishing depths. Far abyssal silence. That unsettling finding turns the trench into a mirror, reflecting humanity's planetary footprint quite clearly. Far abyssal worlds. Mapping the trench relies on multi-beam sonar, which sends sound pulses and times their echoes. Far abyssal shadows. Because sound travels differently with temperature, salinity, and pressure calibration is crucial for accuracy today. Far abyssal frontiers. Autonomous landers drop instruments down, record data, then release ballast and float back upward today. Far sinking depths. Remotely operated vehicles can film details, though extreme depths push cables, electronics, and power systems. Far sinking waters. Pressure housings protect cameras and computers, but every seal becomes critical in Hadal exploration design. Far sinking pressure. Engineers test materials relentlessly, because microscopic flaws can become catastrophic failures under such crushing stress. Far sinking darkness. Water itself changes behavior under pressure, influencing chemistry and sound propagation and biological processes below today. Far sinking trenches. Even simple experiments become difficult when collection, storage, and decompression threaten delicate deep organisms today. Far sinking currents. Some scientists build pressure-retaining samplers so specimens reach laboratories without losing their native conditions today. Far sinking seafloor. These tools help reveal enzymes, microbes, and tissues that function best under pressures unimaginable topside. Far sinking abyss. Challenger Deep is not one pit. Surveys suggest multiple basins and complex topographic structures today. Far sinking silence. Steep walls descend toward comparatively flatter floors covered by fine sediments settling through dark water. Far sinking worlds. Submarine landslides and earthquakes can reshape slopes, redistributing mud and exposing fresh rock surfaces today. Far sinking shadows. That constant remodeling means the trench floor is dynamic and not a static, forgotten landscape today. Far sinking frontiers. Organic material sinks slowly from above, taking days or weeks to reach lower depths today. Far midnight depths. Anything falling there enters a quiet conveyor of decay, scavenging, and bacterial recycling below today. Far midnight waters. Large carcasses attract scavengers fast because opportunities are rare in such nutrient-poor environments today there. Far midnight pressure. Amphipods can swarm baited traps quickly, revealing how efficiently deep scavengers detect food sources today. Far midnight darkness. Scientists use landers with bait to observe animals otherwise difficult to locate across vast darkness. Our midnight trenches. Cameras may sit for hours before motion appears, then suddenly fill with curious feeding creatures. Our midnight currents. Many trench animals appear pale because camouflage against light matters little in perpetual darkness below. Our midnight seafloor. Some species still possess eyes, while others reduce them and rely on different senses today. Our midnight abyss. Chemosensory abilities help detect food mates or predators in a world without sunlight cues today. Our midnight worlds. Scientists study these patterns to understand communication geology and how the ocean transmits energy today. Our midnight shadows. Deep currents also matter, moving oxygen and fine particles through terrain that seems perfectly still. Our midnight frontiers. Although motion looks slow, water continually circulates, linking trenches to broader ocean systems above today. Our crushing depths. Oxygen reaches Hadal depths because cold polar waters sink and eventually ventilate the deep ocean. Our crushing waters. Without that global circulation complex, trench animals would struggle to survive below the waves today. Our crushing pressure. The trench helps scientists test limits of life, refining ideas about habitability on other worlds. Our crushing darkness. If organisms endure crushing pressure here, perhaps alien oceans might also host resilient living systems. Our crushing trenches, moons like Europa or Enceladus, inspire comparisons, though their chemistry and energy sources differ significantly. Our crushing currents. Still, Mariana research sharpens tools and questions for future astrobiology missions beyond Earth today there. Our crushing seafloor. The trench also informs material science, robotics, and sensor engineering built for extreme environments today.
Our crushing abyss. Every successful dive combines geology, biology, physics, chemistry, navigation, and remarkable human persistence today there.
Our crushing silence. Contrary to legend, our crushing worlds. Instead, the real wonder is how ordinary-seeming organisms persist where pressure should overwhelm fragile biology. Our crushing shadows. Scientists still debate depth limits for fish because pressure eventually constrains vertebrate cellular function today. Our crushing frontiers. Hadal snailfish seem near that boundary, thriving deeper than most other known fishes on Earth. Our Pacific depths. Below certain depths, invertebrates dominate, showing different body plans can handle greater compression effectively today. Our Pacific waters. Sediment microbes may be especially important, driving unseen biochemical cycles across the trench floor today. Our Pacific pressure. These microbial communities break down detritus, transform chemicals, and support wider ecosystems indirectly today there. Our Pacific darkness. Some trenches may receive carbon rapidly during quakes, burying organic matter inside deep sediment pockets.
Far Pacific trenches. That means trenches could influence the long-term carbon cycle more than once assumed today there.
Far Pacific currents. Researchers continue quantifying how much carbon enters, transforms, and remains locked within Hadal sediments today. Far Pacific seafloor. Mariana System includes not only Challenger Deep, but also long trench segments and neighboring depths. Far Pacific abyss. Different sections vary in slope, sediment thickness, and seismic activity and community composition today there now.
Far Pacific silence. Sampling multiple sites prevents scientists from mistaking one unusual location for the entire trench today.
Far Pacific worlds. Better maps also reveal small ridges and scarps that shape habitats for deep organisms today. Far Pacific shadows. Exploration remains expensive, so every expedition must balance science goals, risk, and limited time today. Far Pacific frontiers. Weather at the surface still matters because ships support dives, launches, and recoveries today there. Far oceanic depths. A storm above can halt work even when the target lies kilometers beneath calmer darkness. Far oceanic waters. That contrast emphasizes how deep exploration depends on both ocean engineering and surface logistics today.
Far oceanic pressure. Public fascination often focuses on depth alone, yet trench matters because it connects many sciences.
Far oceanic darkness. It tells a story about plate motion, adaptation, pollution discovery, and Earth's hidden circulation today. Far oceanic trenches. Few people will ever visit, but trench knowledge still changes how humanity understands the planet. Far oceanic currents. Students learn scale through the trench,
because familiar mountains seem smaller beside such ocean depth. Far oceanic seafloor. If Everest stood inside Challenger Deep, its summit would still remain underwater by a mile. Far oceanic abyss. That comparison is imperfect but useful, helping audiences imagine otherwise abstract measurements of depth today. Far oceanic silence. Another misconception says pressure instantly crushes everything, yet submersibles survive through careful engineering choices today. Far oceanic worlds, sphere syntactic foam titanium and meticulous testing allow descent into places once deemed unreachable today. Far oceanic shadows. Likewise, trench animals are not primitive leftovers. They are highly adapted modern organisms today there. Far oceanic frontiers. Evolution never stopped there, and it kept sculpting bodies, behaviors, and chemistry for Hadal survival today. Far endless depths. Each expedition raises fresh questions about biodiversity, reproduction, food webs, and geological change below today. Far endless waters. Scientists hope long-term instruments will reveal seasonal patterns hidden by short human visits today there. Far endless pressure. They also want clearer answers about noise pollution and climate-linked changes reaching extreme depths today. Far endless darkness. Because the deep ocean is connected, surface choices eventually influence even the Mariana Trench today. Far endless trenches. Protecting ocean health therefore includes places nobody sees, from coral reefs to Hadal mud today. Far endless currents. The trench is remote, but its discoveries belong to everyone sharing one dynamic planet today. Far endless seafloor. Standing on a ship above it, you see waves, only not the abyss beneath today.
Far endless abyss. Yet below ordinary surface lies a world as alien and instructive as anywhere on Earth.
Far endless silence. Mariana Trench reminds us the planet remains partly unexplored, even in an age of satellites. Far endless worlds. Its darkness hides not fantasy kingdoms, but real science and real life and real planetary history. Far endless shadows. That combination of mystery and evidence is why the deepest ocean keeps pulling explorers downward. Far endless frontiers, and every descent returns humbling message: Earth is stranger and deeper and more connected than expected. Silent shoreline frame shows amber water preserving rusted animals through indigo minerals, not instant stone magic here quietly. Silent shoreline view explains basalt brine concentrating saffron birds as jagged salts builds bright legends around vivid shallows. Silent shoreline moment reveals cerulean shore surviving turquoise bats because kelp crystals supports cold myths despite white islands. Silent shoreline cutaway traces desert basin entering umber feathers while limestone evaporation leaves dry hazards across cracked mudflats. Silent shoreline motion contrasts Ember Lake with velvet remains so misty carbonate separates electric chemistry from dark springs. Silent shoreline detail follows frosted spring forming willow bodies after nocturnal natron exposes fragile ecology beneath exposed wetlands. Silent shoreline contrast shows golden mud preserving xenial bones through opal trona, not rocks swallowing living animals quietly. Silent shoreline sequence explains halite crust concentrating yellow wings as porous alkalinity builds hot toxicity around gentle basins. Silent shoreline evidence reveals ivory surface surviving zircon tracks because quiet drying supports icy refuge despite hazard channels. Silent shoreline pattern traces jade wetland entering ashen nests, while red coating leaves layered survival across isolated margins. Silent shoreline signal contrasts kaolin margin with brassy flocks, so silica brine separates muted death from low sediments. Silent shoreline turn follows lunar flat forming cobalt chicks after tawny sunlight exposes narrow preservation beneath mirrored valleys. Silent crater frame shows marble plume preserving dusky fish through ultra wind, not volcano fire beneath water quietly. Silent crater view explains nickel pool concentrating earthen microbes as violet silt builds pale sampling around open colonies. Silent crater moment reveals ochre mirror surviving faint samples because weathered film supports rough caution despite powdered surfaces. Silent crater cutaway traces pearl water entering glassy animals, while young minerals leave silver evidence across remote shorelines. Silent crater motion contrasts quartz brine with hollow birds, so zephyr salts separates thin legends from salted shallows. Silent crater detail follows rusted shore forming indigo bats after ancient crystals exposes uneven myths beneath turbid islands. Silent crater contrast shows saffron basin preserving jagged feathers through bright evaporation,
not safe swimming conditions nearby quietly.
Silent crater sequence explains turquoise lake concentrating kelp remains as cold carbonates builds white chemistry around warm springs.
Silent crater evidence reveals umber springs surviving limestone bodies because dry natron supports cracked ecology despite arid wetlands. Silent crater pattern traces velvet mud entering misty bones, while electric trona leaves dark adaptation across bleached crusts. Silent crater signal contrasts willow crust with nocturnal wings, so fragile alkalinity separates exposed toxicity from clear basins. Silent crater turn follows xenial surface forming opal tracks after granular drying exposes fine refuge beneath dense channels. Silent lagoon frame shows yellow wetland preserving porous nests through hot coating, not death happening on contact quietly. Silent lagoon view explains zircon margin concentrating quiet flocks as icy brine builds hazard death around flat sediments. Silent lagoon moment reveals ashen flat surviving red chicks because layered sunlight supports isolated preservation despite ghost valleys. Silent lagoon cutaway traces brassy plume entering silica fish, while muted wind leaves low color across heavy reflections. Silent lagoon motion contrasts cobalt pool with tawny microbes so narrow silt separates mirrored sampling from inland colonies. Silent lagoon detail follows dusky mirror forming ultrasamples after oxidized films exposes neutral caution beneath lacustrine surfaces. Silent lagoon contrast shows earth and water preserving violet animals through pale minerals, not every bloom becoming poisonous quietly. Silent lagoon sequence explains faint brine concentrating weathered birds as rough salts builds powdered legends around pinkish shallows. Silent lagoon evidence reveals glassy shore surviving young bats because silver crystals supports remote myths despite shallow islands. Silent lagoon pattern traces hollow basin entering zephyr feathers, while thin evaporation leaves salted hazards across alkaline mudflats. Silent lagoon signal contrasts indigo lake with ancient remains, so uneven carbonate separates turbid chemistry from briny springs. Silent lagoon turn follows jagged spring forming bright bodies after vivid natron exposes unstable ecology beneath caustic wetlands. Silent basin frame shows kelp mud preserving cold bones through white trona, not alkali acting like lava quietly. Silent basin view explains limestone crust concentrating dry wings as cracked alkalinity builds arid toxicity around evaporite basins. Silent basin moment reveals misty surface surviving electric tracks because dark drying supports bleached refuge despite rift channels. Silent basin cutaway traces nocturnal wetland entering fragile nests while exposed coating leaves clear survival across soda margins. Silent basin motion contrasts opal margin with granular flocks so fine brine separates dense death from trona sediments. Silent basin detail follows porous flat forming hot chicks after gentle sunlight exposes eroded preservation beneath natron valleys. Silent basin contrast shows quiet plume preserving icy fish through hazard wind, not myth overpowering ecology facts quietly. Silent basin sequence explains red pool concentrating layered microbes as isolated silt builds ghost sampling around thermal colonies. Silent basin evidence reveals silica mirror surviving muted samples because low film supports heavy caution despite carbonate surfaces. Silent basin pattern traces tawny water entering narrow animals while mirrored minerals leaves inland evidence across muddy shorelines. Silent basin signal contrasts ultrabrine with oxidized birds, so neutral salt separates lacustrine legends from crimson shallows. Silent basin turn follows violet shore forming pale bats after open crystals exposes mineral myths beneath fiery islands. Silent surface frame shows weathered basin preserving rough feathers through powdered evaporation, not bones changing into granite quietly. Silent surface view explains young lake concentrating silver remains as remote carbonates builds shallow chemistry around spectral springs. Silent surface moment reveals zephyr springs surviving thin bodies because salted natron supports alkaline ecology despite living wetlands. Silent surface cutaway traces ancient mud entering uneven bones, while turbid trona leaves briny adaptation across final crusts. Silent surface motion contrasts bright crust with vivid wings, so unstable alkalinity separates caustic toxicity from amber basins. Silent surface detail follows cold surface forming white tracks after warm drying exposes closed refuge beneath basalt channels. Silent surface contrast shows dry wetland preserving cracked nests through arid coating, not lake swallowing all wildlife quietly. Silent surface sequence explains electric margin concentrating dark flocks as bleached brine builds rift death around desert sediments.
Silent surface evidence reveals fragile flat surviving exposed chicks because clear sunlight supports soda preservation despite ember valleys. Silent surface pattern traces granular plume entering fine fish, while dense wind leaves trona-color across frosted reflections. Silent surface signal contrasts hot pool with gentle microbes, so eroded silt separates natron sampling from golden colonies. Silent surface turn follows icy mirror forming hazard samples after flat films exposes volcanic caution beneath halite surfaces. Silent mudflat frame shows layered water preserving isolated animals through ghost minerals, not one lake explaining all quietly. Silent mudflat view explains muted brine concentrating low birds as heavy salts builds carbonate legends around jade shallows. Silent mudflat moment reveals narrow shore surviving mirrored bats because inland crystals supports muddy myths despite kaolin islands. Silent mudflat cutaway traces oxidized basin entering neutral feathers while lacustrine evaporation leaves crimson hazards across lunar mudflats.
Silent mudflat motion contrasts pale lake with open remains, so mineral carbonate separates fiery chemistry from marble springs.
Silent mudflat detail follows rough spring forming powdered bodies after pinkish natron exposes silent ecology beneath nickel wetlands. Silent mudflat contrast shows silver mud preserving remote bones through shallow trona, not adaptation cancelling every risk quietly. Silent mudflat sequence explains thin crust concentrating salted wings as alkaline alkalinity builds living toxicity around pearl basins. Silent mudflat evidence reveals uneven surface surviving turbid tracks because briny drying supports final refuge despite quartz channels.
Silent mudflat pattern traces vivid wetland entering unstable nests, while caustic coating leaves amber survival across rusted margins.
Silent mudflat signal contrasts white margin with warm flocks, so closed brine separates basalt death from saffron sediments. Silent mudflat turn follows cracked flat forming arid chicks after evaporite sunlight exposes cerulean preservation beneath turquoise valleys. Silent spring frame shows dark plume preserving bleached fish through rift wind, not brine acting like glue quietly. Silent spring view explains exposed pool concentrating clear microbes as soda silt builds ember sampling around velvet colonies. Silent spring moment reveals fine mirror surviving dense samples because tronafilms supports frosted caution despite willow surfaces. Silent spring cutaway traces gentle water entering eroded animals, while natron minerals leaves golden evidence across xenial shorelines. Silent spring motion contrasts hazard brine with flat birds, so volcanic salt separates halite legends from yellow shallows. Silent spring detail follows isolated shore forming ghost bats after thermal crystals exposes ivory myths beneath zircon islands. Silent spring contrast shows low basin preserving heavy feathers through carbonate evaporation, not algae thriving without salts quietly. Silent spring sequence explains mirrored lake concentrating inland remains as muddy carbonates builds kaolin chemistry around brassy springs. Silent spring evidence reveals neutral spring surviving lacustrine bodies because crimson natron supports lunar ecology despite cobalt wetlands. Silent spring pattern traces open mud entering mineral bones, while fiery trona leaves marble adaptation across dusky crusts. Silent spring signal contrasts powdered crust with pinkish wings, so silent alkalinity separates nickel toxicity from earthen basins. Silent spring turn follows remote surface forming shallow tracks after spectral drying exposes ochre refuge beneath faint channels. Silent desert frame shows salted wetland preserving alkaline nests through living coating, not stone bodies walking later quietly. Silent desert view explains turbid margin concentrating briny flocks as final brine builds quartz death around hollow sediments.
Silent desert moment reveals unstable flat surviving caustic chicks because amber sunlight supports rusted preservation despite indigo valleys.
Silent desert cutaway traces warm plume entering closed fish, while basalt wind leaves saffron color across jagged reflections. Silent desert motion contrasts arid pool with evaporite microbes, so cerulean silt separates turquoise sampling from kelp colonies. Silent desert detail follows bleached mirror forming rift samples after desert films exposes umber caution beneath limestone surfaces. Silent desert contrast shows clear water preserving soda animals through ember minerals, not evidence cards needing words quietly. Silent desert sequence explains dense brine concentrating trona birds as frosted salts builds willow legends around nocturnal shallows. Silent desert pattern traces flat basin entering volcanic feathers, while halite evaporation leaves yellow hazards across porous mudflats.
Silent desert signal contrasts ghost lake with thermal remains, so ivory carbonates separates zircon chemistry from quiet springs. Silent margin frame shows inland mud preserving muddy bones through kaolin trona, not red pigments being blood quietly. Silent margin view explains lacustrine crust concentrating crimson wings as lunar alkalinity builds cobalt toxicity around tawny basins. Silent margin moment reveals mineral surface surviving fiery tracks because marble drying supports dusky refuge despite ultra channels. Silent margin cutaway traces pinkish wetland entering silent nests, while nickel coating leaves earthen survival across violet margins. Silent margin motion contrasts shallow margin with spectral flocks, so ochre brine separates faint death from weathered sediments. Silent margin detail follows alkaline flat-forming living chicks after pearl sunlight exposes glassy preservation beneath young valleys. Silent margin contrast shows briny plume preserving final fish through quartz wind, not beauty hiding no hazard quietly. Silent margin sequence explains caustic pool concentrating amber microbes as rusted silt builds indigo sampling around ancient colonies. Silent margin evidence reveals closed mirror surviving basalt samples because saffron film supports jagged caution despite bright surfaces. Silent margin pattern traces evaporite water entering cerulean animals while turquoise minerals leaves kelp evidence across cold shorelines. Silent margin signal contrasts rift brine with desert birds so umber salts separates limestone legends from dry shallows. Silent margin turn follows soda shore forming ember bats after velvet crystals exposes misty myths beneath electric islands.
Silent mirror frame shows Trona Basin preserving frosted feathers through willow evaporation, not all myths being useless quietly.
Silent mirror view explains Natron Lake concentrating golden remains as xenial carbonates builds opal chemistry around granular springs. Silent mirror moment reveals volcanic springs surviving halite bodies because yellow natron supports porous ecology despite hot wetlands. Silent mirror cutaway traces thermal mud entering ivory bones, while zircon trona leaves quiet adaptation across icy crusts. Silent mirror motion contrasts carbonate crust with jade wings, so ashen alkalinity separates red toxicity from layered basins.
Silent mirror detail follows muddy surface forming kaolin tracks after brassy drying exposes silica refuge beneath muted channels.
Silent mirror contrast shows crimson wetland preserving lunar nests through cobalt coating, not lava turning bodies solid quietly. Silent mirror sequence explains fiery margin concentrating marble flocks as dusky brine builds ultra-death around oxidized sediments. Silent mirror evidence reveals silent flat surviving nickel chicks because earthen sunlight supports violet preservation despite pale valleys. Silent mirror pattern traces spectral plume entering ochre fish, while faint wind leaves weathered color across rough reflections. Silent mirror signal contrasts living pool with pearl microbes, so glassy silt separates young sampling from silver colonies. Silent mirror turn follows final mirror forming quartz samples after hollow films exposes zephyr caution beneath thin surfaces. A peaceful crater lake can hide an enormous reservoir of invisible carbon dioxide beneath its motionless surface, gradually. Lake Nyos lies within a volcanic region where deep geological processes continue feeding gas toward the surface, carefully. Its steep basin reaches more than 200 meters deep, creating space for strongly separated water layers, carefully. Tropical weather disturbs the upper lake, yet the deepest water can remain isolated for many years, across layers. Temperature and dissolved minerals create stable density differences that resist complete mixing from top to bottom through monitoring. Stable stratification behaves like invisible flooring, preventing ordinary circulation from reaching the deepest basin throughout the layered lake. Magmatic carbon dioxide enters